Plant propagation is the reproduction of new plants from seeds and vegetative parts of the plant, such as leaves, stems, and roots. The two major methods of plant propagation are sexual and asexual propagation. Sexual propagation is the reproduction of plants via seeds. Asexual reproduction produces a plant that is genetically identical to its parent. I'm Dr. DeBusk, and in this video, I'll focus on seed propagation. Sexual propagation is the major means of reproduction in nature and agriculture because it is easy and relatively inexpensive. In a variety of cases, seeds are homogeneous and produce seeds that are true to type, in other words, the same as the parent plant. This is the most common means of propagation for self-pollinating plants. In addition to their expense, some advantages of seeds include that seeds have a convenient method for storage, they remain viable if they are kept cool and dry, and they are an easy method for starting disease-free plants. In some cases, seeds may not be the most practical since it can take a long time for plants to reach maturity. For example, it takes 8 years for pear trees to fruit from seed. This is true for many fruit trees. A variety of factors must be considered for the production and certification of seed. To produce high-quality seed, it is important to choose the proper location. Typically, an arid climate is best because there are fewer disease problems. After a suitable location is selected, you must know when seeds are ripe to maximize germination rates and produce high-quality seedlings. After the seeds are harvested, they should be sieved to remove debris, cleaned, and properly stored to maintain their high quality. Plant breeders select plants with desirable characteristics, such as high yield, disease resistance, and a variety of other traits. Prior to commercial distribution of a new cultivar that a breeder has produced, 4 classes of seed production must occur. 1. Breeder seed, a small amount of seed with desirable traits produced by the breeder. 2. Foundation seed, produced under the supervision of agricultural research stations to assure genetic purity and identity. 3. Registered seed, seed is given to a certified grower for the production. And 4. Certified seed, the progeny of registered seed. After certified seed has been produced, many federal and state laws regulate its packaging and distribution. The law requires that the following information be present on the seed packet: trueness of name, origin, germination percentage, pure seed percentage, percentage of other crop seed and/or weed seed, and percentage of other ingredients. After seeds are stored for any length of time, the germination rate should be tested because this rate decreases with time. Percent germination is the percentage of seeds that will sprout and grow. 90 seeds germinate out of 100 seeds equals 90% germination. If you need 100 plants, you must sow more than 100 seeds. Simply sowing 100 seeds and determining the number of seeds that germinate can easily test the percentage seed germination. This is often done on a moist paper towel. Unfortunately, if it doesn't germinate, it doesn't necessarily mean that it is non-viable because it could be dormant. A more extensive test using tetrazolium can indicate living seeds. Seeds can be planted by direct or indirect seeding. Direct seeding involves planting seeds directly in the soil outdoors. Some examples of crops that can be seeded directly are corn, pea, lettuce, and beans. Small seeds can be pelletized to increase the size to facilitate planting. You see this often with lettuce seeds. Another method, called fluid drilling, uses pre-germinated seeds suspended in a protective gel. The gel prevents seeding injury to the emerging radicle, baby root, and maintains seed moisture. For direct seeding to be successful, a proper site must be selected. The site must be disease-free and have adequate environmental conditions. After the site is selected, you must properly prepare the seedbed by removing any large objects and amending the soil to create proper soil texture for optimal moisture retention and aeration. You must know the optimum date for planting, use correct planting depth and spacing, and make sure seeds receive optimal moisture during the germination process. Indirect seeding involves planting seeds indoors and growing seedlings prior to transplanting to a larger container or to a location outdoors. Some examples of plants that are commonly indirectly seeded are coleus, geranium, zinnia, tomato, pepper, cucumber, and onion. Some of the advantages of indirect seeding are good establishment and less time to maturity, which reduces the actual field growing period. The disadvantages include a higher cost since seedlings are produced indoors, not as easy to plant mechanically and difficult to handle due to bulky vegetative growth, and must be planted before they get pot-bound. Transplanting shock is a major problem with indirect seeding when seedlings are not properly prepared for transplanting and improper planting techniques are used. High-quality transplants start with high-quality seeds. After high-quality seeds have been procured, They need a germination medium that has proper drainage and aeration, adequate water and nutrient holding capacity, favorable pH, proper essential elements, and freedom from insects, weeds, and disease organisms. After a high-quality germination medium has been selected, the proper environmental conditions must be provided to ensure good germination and subsequent seedling growth. The tender seedlings must be protected from damping off, which is a fungal disease that causes the stems to rot at the soil line. The most effective way to control this disease is to use proper sanitation practices and avoid warm, wet mediums. In some cases, when cultural methods are not successful, fungicides are used to control damping off. Prior to transplanting to the field, seedlings should be hardened off to ensure success under harsh outdoor environment. Transplants can be hardened off by gradually subjecting seedlings to cooler temperatures with less frequent watering. For seed germination to occur, the seed must be viable, must be subjected to the proper environmental conditions, and must overcome primary dormancy. There are 5 categories of primary seed dormancy. Physical dormancy, or seed coat dormancy, occurs when the seed covering is impervious to water. Germination can be artificially induced by scarification, which is the process of breaking the seed coat. This can be accomplished artificially either by nicking the seed coat or by soaking the seed in dilute acid for a period of time. In nature, this type of dormancy can be overcome by microorganisms breaking down the seed coat, by the seed passing through the digestive tracts of birds and animals, by mechanical abrasion, by freezing and thawing, and in some species, by fire. The legumes and solanaceous plant families are examples that have physical dormancy. Mechanical dormancy, or hard seed dormancy, is caused by the seed enclosing structure being too strong to permit expansion of the embryo, even though water can penetrate it. Germination can be induced artificially by physically cracking the hard seed coat. In nature, the seed coat can be broken down by microorganisms and/or through the process of freezing and thawing. Walnuts and stone fruits, like peaches, are examples of plants that have this type of dormancy. Chemical dormancy, or inhibitor dormancy, is caused by germination inhibitors which accumulate in the fruit or seed coverings during development. This type of dormancy can be overcome artificially by leaching the seed coat, removing the seed coat, or both. In nature, chemical dormancy is typically overcome by leaching the seed coat. The portulaca and mustard plant families are examples that have this type of dormancy. Morphological dormancy occurs when seeds are shed from the parent plant when their embryos are not fully developed. This type of dormancy can be overcome by treating the seeds with gibberellic acid, a plant hormone used to promote seed germination. In nature, very few seeds will germinate if they are shed from the plant prior to becoming fully developed. The rhododendron and carrot plant families are examples that have this type of dormancy. Physiological dormancy is a general type of dormancy in freshly harvested seeds from herbaceous plants. It is caused by environmental cues such as temperature and light, which lead to an increase in internal growth inhibitors spread throughout the seed and cannot be overcome by removing the seed coat or by leaching, such as chemical dormancy. It can be overcome artificially by stratification, which places seeds on a moist medium at warm temperatures or at cold temperatures, such as for apple, pear, or redbud seeds. The seeds may also be treated with gibberellic acid. In nature, physiological dormancy is overcome by environmental conditions similar to artificially induced stratification. In conclusion, sexual propagation is the major means of propagation in nature and agriculture today because it is easy and relatively inexpensive. You learned important factors involved in the stages of seed production, direct and indirect seeding, and types of dormancy.